So the last time we met, we were talking about photosynthesis. We introduced it briefly. I just wanted to get you familiar with the chloroplast. And that is the location of photosynthesis if you are an autotrophic eukaryote. However, if you are an autotrophic prokaryote and you lack membrane-bound organelles, where do you conduct photosynthesis or metabolic functions? The plasma membrane. The plasma membrane. So if you're a prokaryote, you're a bacteria, and you have no membrane-bound organelles. So you conduct photosynthesis or any metabolic function using your plasma membrane. So we discussed that at the very beginning. We also talked about the general anatomy of a chloroplast. Okay, we talked about the thylakoids being like the little green pancakes. The open area in the middle is the lumen, which is why I have this diagram here, so that you can see an individual thylakoid, and you can see that open area, and again, the term for an open area is lumen. I also mentioned that each of these thylakoids, each individual one, is composed of a phospholipid bilayer. Okay, and I'll kind of expound on that again today. But when you have a stack of thylakoid membranes, we call that a granum. And plural for granum is grana. We also talked about the open space in the chloroplast being the stroma. It's kind of like the cytosol, but cyto is cell, and this is an organelle in the cell, so it's not cytosol, it's the stroma. We also talked about the unique characteristics of a chloroplast, the fact that it has its own genetic material and 2 cell membranes. And what does that give us evidence of, both of those characteristics? At one point in time, it survived on its own. It was a bacteria. What's the theory that explains how it got 2 cell membranes? Endosymbiotic theory. Fantastic, fantastic. Then after we discussed that, we talked about the different pigments. Okay? And I asked, why is the chloroplast green? It absorbs all colors, yet it reflects green. And then we kind of guessed around, what color light do you think it would carry out the best or most photosynthesis? And we guessed around, but we realized after we had our discussion that the least favorite light for a plant is, or a photosynthetic organism is, green. And the reason why is because it reflects green, so it doesn't absorb green. We talked about chlorophyll being a green pigment. Do you recall what the name was for the orangish reddish pigment? Orangish reddish pigment. It started with the C. Carotenoids. Yes, carotenoids. So it kind of looks like carrot, and it's orangish red. Okay, so that kind of helps with that, remembering that. We talked about those 2 pigments. We also talked about the visible light spectrum, that light is not measured in ATP because light is not alive. We measure the energy from light in photons. Fantastic. And the photons, or the wavelengths of those photons, vary in what we see. We can see anywhere from 400 nanometers to 700 nanometers in wavelength, and that's what we call the visible light spectrum. Okay? We also discussed that the shorter the wavelength, like more in the blues and the violets, the stronger and more powerful that photon, versus the reds and the oranges where the wavelength is much longer. There's still energy there, photons, but it's just not as powerful as it is at the 400 end of the spectrum. So that's where we had left off. We just briefly introduced photosynthesis. And here today, I told you I wanted to get into the details. So we're going to begin with photosystems. And again, I put this up here not because I needed you to be able to read all of this, although if you can, that's great. I realize that it's not as clear as I want it to be. But it's so that you could see the actual physical structure of a thylakoid membrane. And you could also see this open area, which we call the lumen. Now, up close, here's the characteristic that I really wanted to point out. So we took right here, and we blew it up. But what is this again that composes the thylakoid membrane? It's a phospholipid bilayer. And if this is a phospholipid bilayer, what is this structure that's here in the middle that's going all the way through? It's a protein. Is this an integral protein or a peripheral protein? Why is it integral? It's going all the way through. All I've done is take information that you already know and have applied it to what we're covering right now. Now, what determines what this protein will do? It's shape, because shape always determines function. So this protein here is called a photosystem. And within this photosystem, I have my two pigments. I have chlorophylls that are represented here in a greenish yellow, and I have my carotenoids that are here in an orangish red. And the whole purpose of this photosystem is to absorb light from the sun. That's its whole purpose. We call this the antenna complex. I'm going to pause for just a second and answer my kid calling. Yes, ma'am. Okay, so did Abby. I'll have to bring it to y'all later. I'm in class. I love you. Thank you. So this area right here that absorbs the photons of sunlight is called the antenna complex. And I have this on the next slide defined, but I just want to kind of show it to you. So antenna complex is responsible for absorbing all the wavelengths of light. That's this area. Down here is our reaction center. And so when I talk about here in just a few moments, the removal or the oxidation of electrons and so on, that's going to occur here in the reaction center. So we have our antenna complex and we have our reaction center. Antenna complex and reaction center, both within this photosystem. But the purpose of a photosystem. There are large complexes of light-absorbing pigments and proteins. So what's their whole purpose? To absorb light. That's their whole purpose. We're going to talk about these two steps. And the first step is literally to take photons and turn them into ATP. And we need the photosystems to do that. Now, here's again this same diagram. But you can see that I have antenna complex defined. And I have the reaction center defined. So that you can have them both in words. So the antenna complex again is filled with the pigments that are responsible for absorbing the different wavelengths of light. And the reaction center is where those electrons are going to be manipulated. Where those electrons are going to be transferred. Now just a reminder, energy cannot be created or destroyed, but it can change forms or it can transform. And how do I get energy to transform? What do I have to move? Electrons. I have to move electrons. That's exactly right. I have to move electrons. So when we talk about oxidizing and reducing, we're moving electrons. And our whole goal here in these photosystems is to take light energy and transform it into ATP. And I'm going to do that by moving electrons. Now the very last point here is that it labels photosystem 1 and 2. I bet you can't guess how many photosystems are in each thylakoid. Just 2. 1 and 2. 1 and 2. And I love the fact that they're named photosystem 1 and 2 because it's pretty straightforward. Photosystem 1 was discovered first. Photosystem 2 was discovered second. Yes, there you go. Random guesses, and we're right. But when I teach you this process, guess which one comes first in the process? 2. Photosystem 1 was discovered first. Photosystem 2 was discovered second. But as we go through the light-dependent reactions, photosystem 2 works first. Photosystem 1 works second. Okay, and I'll keep reiterating that, but I want to go ahead and point that out as we begin. So here again, I have a thylakoid membrane, and I chose this diagram because you can clearly see that it is a phospholipid bilayer. You can clearly see that it is a phospholipid bilayer. You can also see the placement of the photosystems. I have photosystem 2 first and photosystem 1 second. I also have here that photosystem 1 is also referred to as P700 and photosystem 2 as P680. What that means is that's the maximum wavelength of light they can absorb. So photosystem 1 can absorb up to 700 nanometers of light, or a 700 nanometer wavelength. I didn't mean of light, I apologize. And photosystem 2 up to 680 nanometers. Okay, so photosystem 1 is also called P700. Photosystem 2 is also called P680. And I intentionally put this slide together because on your test, sometimes they call them photosystem 1 and photosystem 2, and sometimes they call it P700 and P680, and they don't have photosystem there at all. So I want to make sure that you know what it is. Now, there's one other characteristic of this thylakoid membrane that I want to go ahead and point out, and it's this right here. This is a protein called, and you've already been introduced to this protein before in a previous process, it's called ATP synthase. What does ATP synthase do? What can you tell me about ATP synthase? How do you know it creates ATP? Synthesis means to make or create, okay. What else can you tell me about synthase? It's an enzyme, and you know it's an enzyme because it ends in ase. And all enzymes are proteins. So when you see this, and you see a name that's up close and it ends in ASE, you already know it's an enzyme, and you already know that all enzymes are proteins. And the job of ATP synthase is just what its name indicates. It synthesizes ATP. Now, what's the process of taking ADP and turning it into ATP? Phosphorylation. And we're going to say that again today a few times. Okay? Here is the summary of the reactions that we're about to discuss. I'm going to come back to this, okay? I want to go right here. I made this slide and added it. It is not in the eCampus notes because after I've taught this a few times, I can see where people get confused. So I want to start with just the details, what you need to know. And remember, when you get overwhelmed, when we're studying photosynthesis, the whole goal is to take energy and turn it into food. For photosynthesis, the whole goal is to take energy and turn it into food. What is the goal for cellular respiration? To take food and turn it into energy. Remember that when you're studying. Go back to the big picture. Okay? So I'm going to come over here and I'm going to illustrate to you what occurs during the light-dependent reactions. During the light-dependent reactions. There are two sets of reactions in photosynthesis, light-dependent and light-independent. To begin with, they take place in the thylakoid membrane. So I've already got my little thylakoid drawn here. And I'm going to add my photosystems. So this is PS2, photosystem 2. I'm also going to call it P680. And downstream just a little bit is another photosystem, which is PS1, photosystem 1, or P700. Okay? For the light-dependent reactions, because they're light-dependent, what does that mean they need? They need light. And that makes sense because I have photosystems, and the whole purpose of a photosystem is to absorb light. So I know one of the reactants in this situation is going to be light. Light is going to go in. Excuse me. So I'm going to start here. And I know that one of my reactants is light. So light is coming in. And the only place that it's going to be absorbed are the photosystems. Light is there. Another reactant, something else that goes into the light-dependent reactions, is water. H2O. So we have water and light. And in the light-dependent reactions, just light-dependent, my whole goal is to take light energy, which is measured in what? Photons, fantastic. To take light energy and transform it into ATP, or chemical energy. Okay, that's my whole goal for these reactions here, is to take the energy from the sun and turn it into energy that this photosynthetic organism can use. What's the only way I can transform types of energy? Energy cannot be created or destroyed, but it can be transformed. How do I move it from one type of energy to another? Electrons. So in order to move electrons, I'm going to need an electron carrier. Do you remember what my electron carrier is for photosynthesis? It has NADP. It's the one with the P. So my carrier, my empty taxi, is NADP. I'll put it over here because I'm going to put it here in just a moment. NADP. And then remember I had my ATP synthase right here. So eventually I'm going to also create ATP. But ADP is going to go in. Let me start there. We start with ADP because I'm talking about reactants. So what goes into my light-dependent reactions? I have light, I have water, ADP, and NADP. And for light-dependent, I'm trying to take photons and turn them into ATP, energy that organism can use. Okay? Here's what happens. Sunlight hits photosystem 2. And I have one molecule of water. Can you tell me what happens to water if a photon of light hits it? It's going to split. It's going to evaporate. So when water splits, it splits into hydrogen and oxygen. What does a plant do with oxygen? It's a waste product. So at this point, what occurs is when that photon of light hits photosystem 2, the water molecule is split. The oxygen is released. And then I'm left with my hydrogens. Do you remember what these hydrogens represent? Last week I told you there were different ways that we can interpret electrons. Sometimes electrons are represented by an E, the negative charge, to show you because electrons have a negative charge, or they're represented by hydrogen ions. So what do you think these are representing? The electrons. Okay? So the first thing that happens is that photon of light hits photosystem 2, and it takes this water molecule and splits it into oxygen and hydrogen. So I have my electrons. Do you remember the term for split? We've used this before. When you take something and you split it. I'm going to go back one slide, and I want you to, not right here, this one. What is the word for split? Lysis. What am I using to split this water? Light. What's the root for light? Photo. So when I say photolysis, what does that mean? I'm using light to split water. I'm using light to split this. So this process right here is referred to as photolysis because I used a photon, I used light to split or lyse this water. And when I did that, the oxygen was released, but the electrons, they were excited. So here my electrons are. They're excited. They have a whole bunch of energy. And then they have to travel to photosystem 1. Why do you think I have this line kind of slanting down as that electron travels? What do you think happens to its level of energy? It decreases. Just think about a road trip. When you first leave for the road trip, you're really excited. About 8 hours in, how do you kind of feel? You're like, oh my God, can we stop because I'm going to eat? I just want to go to sleep. Like your energy level drops. Then when you get to the restaurant or you get to the hotel and you re-energize, how do you feel? Excited again. You're like, okay. When these electrons make it to photosystem 1, what's the job of a photosystem again? Absorb light. So what do you think's about to hit this again? Light. So it gets hit again at photosystem 1 with another photon of light. So we have our electrons here. They travel to photosystem 1 where they have lost energy, and they get zapped again. Immediately after they are zapped, these electrons are reduced to NADPH. I need to hold on to these electrons because if I don't hold on to these electrons, I'm going to lose them. So the electrons are added to the electron carrier, and right now I have reduced NADPH. That happens after the second photon hits photosystem 1. What also happens is some electrons feed into, and I put electron and hydrogen so that you could see that I'm talking about the same thing. Some electrons feed into my ATP synthase, and guess what they do? They take ADP and turn it into ATP. What's that process called again? Phosphorylation. Okay. So this is 100% phosphorylation, which means I'm taking ADP and turning it into ATP. But what did I use to phosphorylate this? I did use electrons. How did I get those electrons? Let me throw back real quick. What is oxidative phosphorylation? What did I do in order to phosphorylate in oxidative phosphorylation? I made it smaller. I removed electrons. I did that in cellular respiration. When I had substrate level phosphorylation, what did I do? When I had substrate level phosphorylation in step 1 and step 3 of cellular respiration, I was taking ADP and turning it into ATP, but what was I using? In substrate level phosphorylation. I was using a substrate like glucose. I used glucose to phosphorylate, and in step 3 I used acetyl coenzyme A. So again, my question is, what did I use here to phosphorylate ADP into ATP? What was the main catalyst? What made this whole reaction occur? Light. What is the root for light? Photo. So if you had to guess, where do you think I would put that in reference to phosphorylation? What would you call that type of phosphorylation if I used light? Photophosphorylation. Photophosphorylation is where I used light in order to phosphorylate ADP into ATP. I used light in order to phosphorylate ADP into ATP. Now I'm going to come back over and I want to kind of explain this slide right here. So everything I just drew and illustrated for you is put here in words. Okay? So it talks about the antenna complex being inside the photosystem, and then the reaction center being at the bottom of that photosystem. And it starts off and it says, light strikes P680. What is P680? Photosystem 2, which is the very first photosystem. So light strikes that. And it excites electrons during the process of photolysis. What is photolysis again? Using light to split something. And in this case, we split water. So the oxygens were released and the electrons were harnessed. So it mentions that the oxygen was released. Then those electrons travel down to photosystem 1, which is P700, where they are hit by another photon of light. And when that photon of light hits, it immediately reduces NADP into NADPH, and it helps to phosphorylate ADP into ATP, and that's where this continues. So this is telling you the NADP adds those electrons and becomes NADPH. And the process of taking ADP and turning it into ATP is photophosphorylation. Donovan, what's your question, baby? Correct. Reduce. Reduce is adding electrons in order to make something larger. So everything I illustrated here on the board is put in words on these slides. One thing I want to point out is that this process here is also called the Z scheme. So here's photosystem 2 getting hit by that light, splitting the water, the electron traveling down to photosystem 1 where it is hit again by light, and the NADP is immediately reduced to NADPH, and ADP is phosphorylated into ATP. Why do you think we call this the Z scheme? It's because it looks like a Z. Like that's it. Okay? So the Z scheme is because it looks like a Z. They call it like a sawtooth look. At the end of these light-dependent reactions, so again, my reactants were water, light, ADP, and NADP. My products were oxygen, which was released, ATP, and NADPH. ATP and NADPH are going to go into step 2, which is the light-independent reactions. What I put here on this side was my quick notes as to what happened. So I said a photon strikes photosystem 2, which is P680, and it causes the lysis of water, also called photolysis. The oxygen is released. Those electrons travel to PS1, losing energy. Then a photon strikes PS1, which is also called P700, and immediately those electrons are added to NADP to reduce NADPH. And ADP becomes ATP. It's considered photophosphorylation. Okay? So this is like a summary of all of that together for light-dependent reactions. And where do these occur? In the thylakoid membrane. And the thylakoid membrane is where light-dependent reactions take place. Now, remember how I said there's 2 photosystems, but which one was discovered first? Photosystem 1 was discovered first. Photosystem 1 was discovered first. So it was for a long time that we didn't know about Photosystem 2. So what we used to study is what we call a cyclic electron flow. This still occurs, but at that point in time, we only thought that this occurred, where the electrons followed a cycle as the photosystem was hit by a photon. But the problem is that NADPH is not generated here. When we do the cyclic electron flow, the only thing we make is ATP. It wasn't until later that Photosystem 2 was discovered. And when we realized Photosystem 2 was discovered, we added the process of Photolysis, which gave us the electrons that needed to be reduced down here at that second photosystem, which is technically Photosystem 1. So Photosystem 2 was discovered second. So we have two ways that electrons can flow. The way I taught you initially, which is that Z scheme. It's also referred to as non-cyclic because it's not a cycle. It just goes and it's finished. This second way electrons can flow is what we call a cyclic electron flow, and that's because it flows in a circle, in a cycle. The major difference is if it's following the cyclic electron flow, we're only making ATP. If it's doing non-cyclic, we're making ATP and NADPH. So that you can see the straightforward differences between the two. Questions so far? All right. So then we have our second set of reactions. Our second set of reactions are called the light-independent reactions, and they're referenced as light-independent because they do not need light. They're light-independent because they do not need the light to occur. These reactions are also referred to as the Calvin cycle with a capital C because they're named after the scientist who discovered them. But right here, on the left side, I have some statements that describe these reactions. So I want to discuss and interpret them with you. The first one says, the light-independent reactions, or the Calvin cycles, store some of the energy captured from sunlight in the form of high-energy molecules such as sugar. So what do you think is going to be made in the light-independent reactions? Sugar. Sugar. It mentions that when sunlight is not available, because these are independent reactions, they don't need the sunlight, but they can occur if the sunlight is present. But when the sunlight is not available, stored carbohydrates are broken down by aerobic respiration, which is also our cellular respiration, that takes place in the mitochondria. This is referencing the process that we just finished studying, cellular respiration. Here's my favorite one. Carbohydrates produced in the photosynthetic tissues are shared with the non-photosynthetic parts, such as the roots that cannot conduct photosynthesis. What does that mean? That the photosynthetic tissues that conduct photosynthesis share with the non-photosynthetic parts of the plant. Where's the sugar made? In the leaves. Where are the roots? And why can't they carry out photosynthesis? There's no light. So the part of the plant that carries out photosynthesis creates that glucose or that sugar, and then it sends it down to the roots. Now I have a question for you, and this is going to tie into what I'm going to show you in just a few. If you and I eat fat, our body needs a certain amount of fat. But what happens if we take on too much fat? What does our body do to it? It stores it. When plants carry out, or let me rephrase that, when photosynthetic organisms carry out photosynthesis, they make glucose. But they make way more than they need. So what are they going to need to do with it? Store it. Do you know where they store it? the roots. And do we call it glucose when it's in the roots? No. What do we call it? What kind? It's a complex carbohydrate. It's not glucose anymore, but it's a bunch of glucoses joined together through dehydration. Think about a potato as a root. What do we say potatoes are full of? When you talk about potatoes or corn or onions, they have high levels of starch. Starch is taking a bunch of sugar and sticking them all together through dehydration. So the plant makes all that, I said plant again, but it doesn't have to be a plant. It's just in this case it is. But the organism makes a bunch of glucose and then it stores it so that it can share it with the plant or that organism, that photosynthetic organism, whenever there's not resources available. So the second part here says, during the Calvin cycle, CO2 is reduced. So is CO2 getting bigger or smaller if it's reduced? Bigger. Oxidized means it's getting smaller. Reduced means it's getting bigger. CO2 is reduced and converted into organic substances. I don't know why there's a big space there. But what is CO2 turned into? That would be making it smaller. If carbon dioxide was turned into carbon, what is an organic substance? It has carbon in it, but what's an example of an organic substance? All organic means is it has carbon. What, in this process of photosynthesis, what are we going to take CO2 and reduce it to? Just a hypothesis. If you have the equation, you already know the answer. It's glucose. We're going to take CO2, carbon dioxide, and we're going to add to it until it gets big enough to become glucose. That's what happens in the light-independent reactions. So, quick summary, the light-dependent reactions, we take photons, light energy, and we turn them into ATP, or chemical energy. And the light-independent reactions, we're going to take that CO2 and we're going to reduce it into glucose. We're going to take carbon dioxide and we're going to reduce it into glucose. Okay? The process of reducing CO2 to glucose is called carbon fixation. You're going to see the term fix. Has this carbon dioxide been fixed? So that means, has it been reduced? And I'm going to explain what I mean as I move forward. In order for these reactions to take place, we're going to need an enzyme. The enzyme we're going to use is Rubisco. Why is this not a common name for an enzyme? What is it lacking? ACE. So a lot of people skip over it and think, oh, well, that's not an enzyme because it doesn't end in ACE. Not all enzymes have to end in ACE. It's just that the majority of them do. So Rubisco is the enzyme responsible for taking CO2 and reducing it into glucose. Rubisco is the enzyme that's responsible for taking CO2 and reducing it into glucose. There's a couple things I want to point out on this slide, and then I want to go to demonstration mode again. So the Calvin cycle continues to spin, and that's probably why it's called the Calvin cycle. Okay? We need one carbon dioxide molecule for each turn, for each spin. How many carbons do I need for glucose? How many carbons are in glucose? What's the molecular formula for glucose? C6H12O6. Fantastic. So in order to get 6 carbons, I am going to need 6 carbon dioxides. Does that make sense? Because there's 1 carbon in each carbon dioxide. So that cycle is going to have to go around 6 times. This cycle has to go around 6 times in order for me to take 1 carbon dioxide and turn it all the way into glucose. There are 3 steps for this, but I want you to understand what these terms are. So we're going to use the term PGA. PGA, and it was just given to you, but I wanted you to see what it was an acronym for. It stands for 3-phosphoglycerate. PGA. G3P is glyceraldehyde 3-phosphate. So coming back over to our diagram, can you tell me what's going to be coming in to the light independent reactions? Carbon dioxide. No light, because these are light independent. What were my products here? ATP, I'm going to need energy. What else am I going to need to make carbon dioxide bigger? Plants release oxygen. Photosynthetic organisms release oxygen. Hydrogen, but what do we call those? We don't call them hydrogens, we call them electrons. And what's carrying my electrons? My electron carrier, which happens to be this is what goes in. These are my reactants for the light independent reactions. These reactions do not take place in the thylakoid. They take place in that open space. Do you recall what that open space is called? Stroma. Yes, these reactions take place in the stroma. That's the location of these. And once again, the big picture of the light independent reactions is to take CO2 and reduce it into glucose, C6H12O6. That's the goal of the light independent reactions. Yes, sir. The goal of the light independent reactions. Now, what does reduce mean again? It gets bigger. So I'm going to start off with carbon dioxide. And then as this process spins, as the Calvin cycle is carried out, CO2 is converted to 3-PGA. To 3-PGA. You do not have to understand how I got to that. You just have to know what it was converted to. Then it's further reduced to G3P. So I'm taking all these electrons and I'm adding them as we continue to spin around. So I go from CO2. My first intermediate is PGA. As I continue to add, it becomes G3P, and G3P will ultimately become glucose. G3P will ultimately become glucose. CO2 enters, and as the Calvin cycle is carried out, it becomes PGA. It continues to be conducted. We continue to further reduce it into G3P, and eventually, after it has turned 6 times, it becomes glucose. The process of taking CO2 and reducing it to glucose is called fixation. Carbon fixation. You might also hear it called fix. Like, how do we fix this? And the enzyme that carries out these reactions is called Rubisco. The enzyme that catalyzes these reactions is called Rubisco. The enzyme that catalyzes these reactions is called Rubisco. What does catalyze mean? It doesn't mean breakdown. To initiate or to get it going. Huh? Oh, it's all right. I said the wrong thing all the time. But to catalyze means to initiate or to get it going. So, for the light-independent reactions, here's our summary slide. The location is the stroma. My reactants are the ATP and NADPH that were provided from the light-dependent reactions, and then carbon dioxide. My product is going to be glucose, then the ADP and NADP that need to be recharged. Because I used these here, I need to recharge them again in the light-dependent reactions. So I have the intermediates here. So CO2 is initially it needs to say PGA, to 3-PGA, and then to G3P, and then ultimately it becomes glucose. The enzyme that carries this function out or catalyzes this reaction is Rubisco. What questions do you have? Did you have one? So this is the summary of this. Go ahead.
So light-independent reactions and light-dependent reactions work in a cycle like cellular respiration and photosynthesis?
Yes. I'll show you what I'm talking about here in just a second. So real quick on Rubisco. It says Rubisco is the enzyme that catalyzes the fixation of carbon dioxide to glucose. So I've already said that. Rubisco provides the source of organic molecules for most of the world's organisms. I need you to explain to me what that means. Rubisco provides the source of organic molecules for most of the world's organisms. It doesn't provide carbon dioxide. What is it allowed to be turned into? Sugar. Rubisco can take our waste, carbon dioxide, and it can make it into food. Rubisco is an enzyme that can take our waste from our vehicles, waste from our body, and turn it into glucose, food. And what type of organisms need glucose? All living organisms. Guess what you need? Do you know what happens when your glucose levels get low? You faint. Your brain will shut off. If you continue to go without it, you will expire, 100%. But if your blood glucose levels drop, you faint. Your body hits a kill switch and says, your brain cannot function without glucose. If you have a hunger headache, it can usually be remedied with a little bit of chocolate because it's got quick glucose, and that's what your brain needs. All organisms, all living organisms need glucose. Rubisco is the enzyme that can take carbon dioxide and turn it into glucose. And what organisms need glucose? All living organisms. So that's what it's meaning right here. It says, on average, about 100 billion tons of carbon dioxide are converted into carbohydrates every year, just by this enzyme, because we have that many photosynthetic organisms. Then it says, and this is the part I really want you to think about, Rubisco represents 50% or more of the total protein in leaves. What does that mean? Rubisco represents 50% or more of the total protein in leaves. It's an enzyme, and all enzymes are proteins. There's a whole lot of Rubisco in the leaves of photosynthetic organisms. Why in the leaves? That's where photosynthesis takes place. Yes. This is a diagram of Rubisco, that enzyme. Is this a random shape? No. It's 100% intentional. There's no random aspect to that at all. So, additional photosynthetic products. I touched on this a little while ago, and I said I'll get to it in a few slides. Plants make extra glucose. Photosynthetic organisms make extra glucose, and what do they do with it? They store it. So the correct answer for photosynthesis is yes, it makes glucose. But what happens, what do we call it when we put two sugars together? Disaccharide. What type of reaction puts two sugars together? Where I have two and I put them together. It's not catabolic. Catabolic would be breaking it down. It's anabolic reaction. Is it dehydration or hydrolysis? Dehydration. Throwback right here. What type of bond is created when I put two carbohydrates together? Glycosidic. Fantastic. Fantastic. Like if I was a gymnast or I could do a flip without hurting myself. I can't even do a flip. And anyway. I'm excited right now, Erica. Thank you for that. So this is what this diagram is showing you. That it doesn't just, when we say it makes glucose, yes. But it has the ability to make sucrose, which is a disaccharide, up to very complex starches. So recap. And this is where I was coming back to what I was referencing. This was a slide that was previous in this slide deck. And I told you I would come back to it as a summary. So the light-dependent reactions take energy from the sunlight and convert it into chemical energy, or ATP. And we also grab those electrons with our electron carrier, which is NADP. We reduce it to NADPH. Those reactions can be either non-cyclic, which is the Z scheme, or cyclic. If it's only cyclic, it makes only ATP. And if it's the Z scheme, it makes ATP and NADPH. These reactions occur in the thylakoid. And again, the products are ATP and NADPH. The second set of reactions for photosynthesis are called the light-independent reactions, or the Calvin cycle, with a capital C. What happens here is we take that excess energy and we build high-energy sugar. What's a high-energy sugar? Glucose. These reactions are also called the Calvin cycle. I've already mentioned that, named after the scientist. They occur in the stroma, which is that open area. They use electrons to produce glucose from carbon dioxide. What's the process of taking carbon dioxide and turning it into glucose called? Fixation. What enzyme is used to fix carbon dioxide? Rubisco. So this is an overall summary of both sets of reactions. This slide is also being reintroduced from this slide deck. So, inside my chloroplast, I have my thylakoid membranes, which is where my light-dependent reactions occur. For my light-dependent reactions, big picture is I'm taking photons and turning them into ATP. So I'm just converting forms of energy. And you convert forms of energy by transferring electrons. So I have water that comes in here and gets lysed, so photolysis, so that oxygen is released. Those hydrogens, which are electrons, are used to photophosphorylate ADP into ATP, as well as reduce NADP into NADPH. That occurs in the light-dependent reactions, which specifically take place in the thylakoid membrane. The products of the light-dependent reactions go in to the light-independent reactions. So the ATP and the NADPH go here so that they can be used to fix carbon dioxide into glucose. The enzyme that's responsible for fixing carbon dioxide into glucose is Rubisco. When I use that ATP, I release it as ADP and send it back over here to get recharged. When I use those electrons on that electron carrier, it comes over here to pick up more electrons. On the Calvin cycle, or the light-independent reactions, when can they occur? Anytime, day or night, because they do not need light. The light-dependent reactions require what? Light. So that summarizes the light-dependent, light-independent reactions of photosynthesis. What questions do you have? I have one more topic that we have to discuss, because what if plants don't breathe in carbon dioxide? And I had talked about at the beginning, our plants in Texas are quite unique, some of them. The topic is photorespiration. So what does photo tell you? Light. And respiration tells you, though, that we're taking in what gas? Oxygen instead of carbon dioxide. Let me explain the relationship between oxygen and carbon dioxide and a photosynthetic organism. I'm going to use myself. I know what I should eat. I know what type of foods make me feel good. And once I eat them, mentally, my blood sugar stays pretty balanced. I'm good. My endurance is good. I know what good foods are, and I meal prep because of that. However, sometimes I leave home and I forget my lunch, or I forget whatever it was. I was just going too fast. And what do you think I do on those days? I usually go to the gas station because I'm like, okay, I'm hungry. But the gas station doesn't have my meats all prepped or anything like that. As soon as I walk into the gas station, I only have one snack in mind. And you're probably going to judge me right now, and that's okay. But I'm a fan of Sour Patch Kids. And I don't care if I buy the small little snack one or the 1 pound bag. Whichever size I buy, as soon as I open it, I eat them all. It's just like for whatever reason, I cannot stop. But I also know exactly how I feel after I'm done eating those. It hurts my mouth because it was so sour. Like it burns my mouth. And then my energy levels are high for a couple minutes, and then I start to kind of shoot down. Even though I know that, guess what I will still do? I will still buy them every single time. Every single time. And I even know it when I leave the house. I'm like, dang it, I forgot my lunch. And I already have in my head, I guess I'm going to have to get Sour Patch. And I know I'm going to feel like crap. I know I'm not going to want to wake up and work out the next morning either because I'm going to be so tired. But I still do it. Plants, or photosynthetic organisms, carbon dioxide is what allows them to run efficiently. They need carbon dioxide to do what they do. However, oxygen is kind of like their Sour Patch Kids. They know they shouldn't use it, but they like it. When plants have access to oxygen, they will photorespirate, which will eventually lead to their death. Just like if I ate Sour Patch Kids every single day, it would eventually lead to problems and long-term expiration. Plants, or photosynthetic organisms, photorespirate when their stomata are closed. And we talked about this the other day. In Texas, because it gets so hot, our plants are in risk of desiccation or dehydration, which means they're going to dry out. So instead of leaving their pores open so that carbon dioxide can come in and oxygen be released, they close their pores. So closing their pores keeps all the water in, but it also doesn't let the oxygen that it's making out or the carbon dioxide in. And so as the plant is releasing oxygen into the leaves, it can't escape. So it actually starts using its own waste products to carry out the process of photosynthesis. And what do you think if you use your waste products for nutrition? Tell me where you think that's going to go. It's not going to be a good thing for you. So I found this diagram that shows you this. Here's photosynthesis. The organism is happy because it's using carbon dioxide. Rubisco is helping it to convert it into glucose. Here is an organism, a photosynthetic organism that is photorespirating, which means instead of taking in carbon dioxide, it is taking in oxygen. And look at how it's making this plant feel, or this photosynthetic organism feel. That's a mad face, right? It's not happy. So I found this as well that kind of summarized, well, why would an organism that could carry out photosynthesis decide to photorespirate? In good conditions where photosynthesis occurs, the temperatures are pretty moderate or low. There's a lot of carbon dioxide and not so much oxygen. So the plant can then conduct photosynthesis, or that photosynthetic organism can conduct photosynthesis, versus a very high temperature where the plant or organism is at risk of desiccation or drying out. And there's only a little bit of carbon dioxide, but there's a lot of bit of oxygen. And so it's like, well, just might as well have a snack. And you can't just eat one Sour Patch Kid. You have to eat a few. And then it turns into the whole thing. So, long term, if organisms that are photosynthetic continue to photorespirate, they will expire. However, there are some organisms that have adapted and now evolved processes where they can overcome those conditions in order to stay alive. And organisms in Texas are one of them because of our extreme heat. So, the terms are C3 and C4 photosynthesis. C3 is what we have discussed this entire time. Your houseplants carry out C3 photosynthesis, which means when they carry out the Calvin cycle, they make 3 PGA. We've said that. They make 3 PGA. That's a 3-carbon molecule. Okay? So C3 plants, those are your house plants and so on. If you take your house plants and you set them out on your porch in August, can you tell me what's going to happen to them? They're going to die. They're going to die. You can't do it. They cannot withstand that heat. C3 plants cannot withstand that heat. However, in areas like ours, there are many plants that are considered C4 plants. What C4 plants can do is instead of making a 3-carbon intermediate, where they have just enough carbon to conduct photosynthesis, this organism has the ability to create a 4-carbon intermediate, which means it can hold just a little bit extra carbon. So when the carbon dioxide levels are low, it can throw those carbons out a little at a time so that that plant can continue to carry out photosynthesis without using oxygen or conducting photorespiration. C4 avoids photorespiration because photorespiration, again, long term, is lethal to a plant or a photosynthetic organism. So I wanted to cover what the difference is between a C3 and a C4, and the difference is in a C3 plant, they make PGA. In a C4 plant, they make a 4-carbon intermediate called PEP, which is phosphophenolpyruvate. PEP. And I mentioned when I discussed this at the beginning of this chapter that a lot of these organisms open and close their stomata, their pores, at night because they don't want to dry out. So when we look at this type of photosynthesis, I'm going to come back to this slide. Here's what you and I have studied so far, C3. Okay, we have our light-dependent reactions that occur, and then the Calvin cycle are light-independent reactions, and it makes glucose. In a C4 plant, instead of making a 3-carbon intermediate, the PGA, it makes a 4-carbon intermediate. Outside, in this area, they occur at different locations. So instead of the entire cycle happening at one spot, it begins here, and then it ends right here, outside of this bundle. We call this the bundle sheath, because this are the vessels. So the bundle sheath, this is the wrapping around those bundles in yellow. So we create the PEP, that extra carbon dioxide is the extra carbon is kind of grabbed here, and then it will make its way here, further inside, where it will conduct the Calvin cycle. The reason it goes further inside is because it's harder for oxygen to get to this tissue. So it kind of says, okay, I'm going to start here, and I'm going to go to a secret hideout so that I can carry out this process. So C4 plants carry this out at different locations. Then there's CAM plants, and they are CAM plants, I need to say that. And CAM, I have this on a slide, stands for Chloroplast acid metabolism. You don't have to know that. But instead of at different times, they start out at night, and they finish during the day, and then they open up at night, and they close during the day, and so on. So C3 carries out normal photosynthesis. C4 carries out the different reactions at different locations. And CAM plants carry them out at different times, so during the night and day. Okay, so I'm going to go back. C4 plants use PEP, which is that 4-carbon intermediate, and they do this to avoid photorespiration, so that photorespiration is minimized, because we don't want our plants or our photosynthetic organisms to utilize oxygen. It says that these plants are more efficient at higher temperatures. Only. If I take these plants to Alaska, are they going to do well? No. If I take these plants just a few states north, are they going to do well? The answer is no. They need to be in extreme heats. So here's showing you that in C4, they takes place in different locations. So it starts here, and then it will finish here, around that bundle sheath. Again, this is to allow for extra carbon dioxide to kind of be harnessed, and then O2 is not used, and the plant avoids photorespiration. In CAM photosynthesis, it occurs at different times. I put on this slide what CAM stands for, Chloroplast acid metabolism. And these carry out part of their cycle at night, and then the other part during the day, so they do it during different times. Again, both of these are adaptations that plants have evolved from so that they could avoid photorespiration, so that they could continue to carry out photosynthesis using carbon dioxide instead of oxygen. Questions here?
So the C4 plants use the PEP
Instead of PGA. Yes, ma'am. Yes, ma'am. Okay, that's the end of. Oops.